In this video, we will discuss about DNA and RNA. DNA and RNA is two types of nucleic acid that will enable the living organisms to reproduce their complex components from one generation to the next. The DNA provides direction for its own DNA replication, and also it will direct the RNA synthesis through RNA and control the protein synthesis. This entire process is called gene expression. So from this diagram, we can assume that the DNA will undergo DNA replication and the DNA will undergo transcription to produce mRNA, and mRNA will undergo translation to produce polypeptide or proteins. DNA is double helix joined together by sugar phosphate backbone and hydrogen bond along the polynucleotides, while RNA consists of single strand of polynucleotide, and the nucleotides may form hydrogen bond at specific segment of the polynucleotide. Nucleic acid are macromolecules that exist as polymers that called polynucleotide. Each polynucleotide consists of monomers that called nucleotide. A nucleotide composed of three components: the 5-carbon sugar, nitrogen-containing base, or also known as nitrogenous bases, and phosphate group. This is the basic components of nucleotide. It consists of sugar, phosphate group, and nitrogenous base. Another example of nucleotides. For this one, it only have one phosphate group, and the next nucleotide, it consists of two phosphate group. Generally, all of the nucleotide consists of the same components: pentose sugar, phosphate group, and nitrogenous base. The structure of nucleotide. Polynucleotide, it consists of nucleotides that join together by phosphodiester bond, and the monomer for the polynucleotide is nucleotide. The nucleotide, it consists of phosphate group and nucleoside. Nucleoside, it consists of nitrogenous bases and sugar. For the nitrogenous bases, it consists of pyrimidine, thymine in DNA, and uracil in RNA. For purine, it consists of adenine and guanine. For the sugar, in DNA, it will be called deoxyribose, and in RNA, it will be called ribose. The 5-carbon sugar or pentose sugar, it consists of deoxyribose in DNA and ribose in RNA. The difference between ribose and deoxyribose is lack of hydrogen atom at carbon number 2 in deoxyribose. The nitrogen-containing base or nitrogenous base. There will be two types of nitrogenous bases: pyrimidine and purines. Pyrimidine, it consists of one 6-membered ring of carbon and nitrogen atoms. For example, cytosine, thymine in DNA, and uracil in RNA. For purine, it consists of 6-membered ring fused with 5-membered ring of carbon and nitrogen atoms. For example, adenine, it consists of 6-membered ring fused to 5-membered ring. And the example of purine is adenine and guanine. You can use this acronym PYSCUT to explain pyrimidine. PY, it stands for pyrimidine. S, it stands for single or one 6-membered ring of carbon and nitrogen atom. C, it stands for cytosine. U for uracil, and T for thymine. For purine, you can use this acronym PUDAG. PU, it stands for purine. D, it stands for double or two 6-membered ring fused with 5-membered ring of carbon and nitrogen atoms. A, it stands for adenine. G for guanine. Adenine, guanine, and cytosine can be found in DNA and RNA. but thymine only can be found in DNA and uracils only can be found in RNA. The formation of polynucleotide. The nucleotides are combined together by condensation reaction to form polynucleotide. During the condensation, nucleotides are joined by phosphodiester bond between the phosphate group of the fifth carbon and hydroxyl group of the third carbon of the other nucleotide. The repeating sugar phosphate unit will form the backbone that we call sugar phosphate backbone or phosphodiester bond. One end has a phosphate attached to 5' carbon and the other end has hydroxyl group on a 3' carbon. At the 5' end, we have the phosphate group, and at the 3' end, we have the hydroxyl group. And in between of the nitrogenous bases, thymine and adenine, we have this hydrogen bond, 2 hydrogen bond between thymine and adenine, and between guanine and cytosine, there will be 3 hydrogen bonds. So this is the polynucleotide or nucleic acid in DNA. Next, the formation of polynucleotide. First, the covalent bond will form between the nitrogenous base and the pentose sugar. The second bond is hydrogen bond. The hydrogen bond will form between the nitrogenous bases. And next, we have the phosphodiester bond. The phosphodiester bond will form between the phosphate group and pentose sugar. So this is the covalent bond, and here is the hydrogen bond, and here is the phosphodiester bond. Next, the structure of DNA based on Watson and Crick model. Generally, the DNA consists of 2 strands that we call DNA double helix. One strand run from 5' to 3', and the other strand run from 5' to 3' in opposite direction. This is what we call anti-parallel. And in between of the nitrogenous bases, we have the hydrogen bond. And the nucleotides will be held together by phosphodiester bond that form between the pentose sugar and phosphate group. So here is the description. James Watson and Francis Crick proposed the double helix model in 1953. According to Watson and Crick, the 2 sugar phosphate backbones run in opposite direction, 5' to 3' direction from each other. This is what we call anti-parallel. The sugar phosphate backbones are on the outside of the helix. The nitrogenous base appeared in the interior of the helix. The sugar phosphate backbones held by phosphodiester bond. The 2 strands are held together by hydrogen bonds between the nitrogenous base. The double helix is compatible or complementary to each other. Adenine in one strand will pair with thymine in the other strand. Guanine will pair with cytosine, and the rule is adenine must pair with thymine with 2 hydrogen bonds, and guanine must pair with cytosine with 3 hydrogen bonds. In DNA, the amount of adenine always equal to the amount of thymine, and the amount of guanine always equal to the amount of cytosine. This is according to the Chargaff's rules. If one strand has the sequence of 5' AGG TCCG 3', the other strand or the complementary strand must have the sequence 3' TCC AGG C 5', and this is what we call anti-parallel between the DNA double helix. The adenine will pair with thymine with 2 hydrogen bonds, and guanine will pair with cytosine with 3 hydrogen bonds. There will be 3 types of RNA, mRNA, tRNA, and rRNA that consist of large and small ribosomal subunits. The function of RNA. Messenger RNA or mRNA will carry the genetic code transcript. tRNA or transfer RNA will transfer the amino acid to the ribosome during protein synthesis. And third, we have rRNA or ribosomal RNA consists of large and small subunit. It will facilitate the specific binding or coupling of tRNA with mRNA. Compare between DNA and RNA. DNA basically consists of double-stranded, while RNA is single-strand. The nitrogenous bases for DNA consist of cytosine, guanine, adenine, and thymine, while for RNA, the nitrogenous bases consist of cytosine, guanine, adenine, and uracil. The difference is thymine in DNA and uracil in RNA, or the other nitrogenous bases are the same in both DNA and RNA. In terms of pentose sugar, in DNA, it will be deoxyribose, and in RNA, it will be called ribose. Comparisons between DNA and RNA. For the similarities, the monomer for DNA and RNA is nucleotide. Both are nucleic acid or polynucleotide, and both will have sugar phosphate backbone. For the differences, in your exam, you are advised to write your answer in paragraph form, and you must use conjunctions such as while, whereas, but, to differentiate between DNA and RNA. For example, DNA consists of two polynucleotides, while in RNA, it consists of one polynucleotide or single-stranded polynucleotide. The pentose sugar in DNA is deoxyribose, while in RNA, the pentose sugar is ribose. For the nitrogenous bases, in DNA, it consists of adenine, thymine, guanine, and cytosine. We must mention all of the nitrogenous bases involved. While in RNA, it consists of adenine, uracil, guanine, and cytosine. In terms of size, DNA is larger molecular size, while RNA is smaller molecular size. DNA is stable molecule, while RNA is less stable molecule. DNA consists of only one type of DNA, while RNA consists of three types: mRNA, tRNA, and rRNA. DNA is susceptible to UV damage, while RNA is less susceptible to UV damage. The function of DNA is to carry genetic information, while RNA involved in protein synthesis. That's all for now, and hope to see you again in the next video. Thank you.